Welcome to Chem Help ASAP. This video describes the discovery of IPG7236. This compound is an antagonist for CCR8, a target with potential use in oncology. The video reports the discovery program as described in a Drug Annotation article in the Journal of Medicinal Chemistry by scientists at Immunophage Biotech in Shanghai. The citation is at the bottom of the screen, and a link to the article can be found in the video description. This video does not describe every aspect of the article. I've selected the parts I consider to be the highlights that best summarize the project and fit within the courses I teach. If you happen to be a scientist who worked on this project and are at liberty to speak about this research or your general work at Immunophage, I would be very interested in interviewing you for a separate channel video. The channel email address is in the video description. The target for this campaign is CCR8. CCR8 stands for CC, that's cysteine, motif, chemokine receptor 8. CCR8 is a GPCR. CCR8 has multiple endogenous ligands, one of which is thymus activation regulation cytokine, TARC. CCR8 is upregulated in tumor-resident Treg cells, and activation of CCR8 is associated with immune escape. Well, this is a problem. Treg cells in tumors have high levels of expression of a receptor that allows both evasion of the immune system and unchecked tumor growth. Therefore, the plan of this program is the design of a CCR8 antagonist that can block the action of CCR8, reduce immune escape, and hopefully result in slowed tumor growth. Other researchers have pursued CCR8 antagonists. Selected examples, which are also in the article, are on the screen. LMD-D from Millenium, SB649701 from GSK, and AZ084 from AstraZeneca. Despite promising potency, these compounds all have a range of issues: poor solubility, low membrane permeability, and poor bioavailability. Regardless of the liabilities of these compounds, the team at Immunophage chose to work within this compound set to see if the properties within this series might be improved to achieve promising in vivo efficacy. Ultimately, the team selected LMD-D as its starting point. It is possible that other compounds were explored, but the paper only discusses LMD-D. The compound has three main parts: the aromatic amide on the bottom left, the naphthalene core, and this urea side chain. The compound suffers from both solubility and PK issues, so there was no screening campaign. LMD-D is the initial lead. The potency is fine, but its ADME properties need improvement. Before we jump into lead optimization, let's introduce the key assays that will assist the researchers. First is the TANGO assay. TANGO is a cell line by which the CCR8 antagonism is measured. Activity is read as an IC50 value. For the ADME properties, membrane permeability was monitored with CACO2 cells. The assay determines permeability from the apical to basolateral face of the membrane, A to B, as well as the permeability in the opposite direction, B to A. These are the apparent permeability of the compound. The ratio of these two numbers is the efflux ratio, ER, which is a dimensionless number. Finally, solubility is a challenge in many of these CCR8 antagonists, so a solubility assay is also key for this program. Let's see some analogs. The first step was to work on the urea side chain in the lead, this R group, off of the nitrogen on the far right. The original R group, the urea side chain, is shown in the first entry of the table. As you can see, the potency of LMD-D is excellent with an IC50 value of 4 nanomolar. The team looked at a number of aryl and acyl groups on the nitrogen, but potency was very poor. Switching to an alkyl group with a tethered nitrogen regained some potency. In the end, this alkyl substitution with an N-methylpiperidine gave the best potency in compound 22, which is actually a bit more potent than LMD-D. Moving to the naphthol ring, we have three compounds on this slide. The first is 22, which has the original naphthol ring. You can see the excellent potency, but apparent permeability is rather low and solubility is terrible. Trimming the naphthol ring to a single aromatic ring with a methyl group loses some potency, but greatly improves solubility, which is vital for an orally administered drug. Adding another chlorine on the ring restores all the lost potency. The solubility is still good, but the efflux ratio rises dramatically. A high efflux ratio is often an indicator of poor oral absorption. Therefore, although compound 1 is not as potent as 30, the very high efflux ratio of 30 caused it to be disfavored relative to compound 1. There is a third part to these molecules that can be optimized. That is the aromatic amide on the far left. As it turns out, attempts to modify this group did not improve the overall properties of the series, so this group was left intact, and compound 1 emerged as the best compound in the lead series. This screen highlights the in vitro ADMET properties of 1. The C log P is low at just 2.6. Plasma protein binding is around 64%. Microsomal stability is excellent. Stability against hepatocyte metabolism is a bit lower, but still outstanding with a half-life of 522 minutes. Microsomal stability is an indication of phase 1 cytochrome P450 oxidation, and hepatocyte metabolism shows both phase 1 and 2 metabolic reactions. So it seems that any observed hepatic clearance of compound 1 will be mostly through phase 2 reactions, not phase 1 redox reactions. Regardless, hepatic clearance should be low. Compound 1 shows no safety risks from hERG inhibition, CYP inhibition, or other common safety panel assays. Here are some in vivo PK data. Compound 1 shows fairly uniform behavior across all 4 common preclinical species. Note the very satisfactory bioavailability values. These results are encouraging given the ADME challenges seen in previous reported CCR8 antagonists. The paper does not disclose the half-lives, but the half-lives can be estimated from the provided clearance and volume of distribution values. The observed half-life values are somewhat short across the board. This observation is surprising given the high in vitro metabolic stability of compound 1. It is possible that compound 1 undergoes extensive renal excretion given its very modest membrane permeability. The authors, however, do not mention if the drug is found unchanged in the urine. With the PK data in hand, let's see compound 1's in vivo efficacy. The data in this table come from the supplementary information and are shown in Figure 3 in the paper. Compound 1 was dosed over a 21-day period to mice with a human breast cancer. Dosing was oral and twice per day, perhaps required to achieve adequate exposure because of the short half-life of compound 1. Compound 1 does show a dose-response effect for inhibition of tumor growth in this animal model. The researchers analyzed the tumor tissue from the previous study and recorded 2 notable observations. First, the administration of compound 1 reduced the presence of CCR8 expressing Treg cells at higher doses. Second, the administration of compound 1 increased the presence of CD8 T cells at higher doses. Recall back to an earlier slide that antagonism of CCR8 is associated with immune escape within the tumor environment. Treatment with compound 1 reduces the Treg cells that lead to immune escape with a corresponding increase of T cells that can mediate an immune response. Of course, the previous slide adds to this picture because the tumors are observed to grow more slowly in the high-dose groups. All these data are consistent and encouraging. The researchers also performed a combination treatment study with an anti-PD-1 antibody a standard treatment for certain cancers. The study was shorter than the previous one. Compound 1 was again orally dosed twice per day. The combo treatment group showed greater inhibition of tumor growth than either of the monotherapy groups. We have seen that compound 1, also known as IPG7236, shows in vivo efficacy in animal disease models for breast cancer. No notable safety risks were reported. Although not mentioned in the paper, IPG7236 is being studied in a phase 1 clinical trial. The clinicaltrials.gov identifier is on the slide. The study is for IPG7236 alone, with twice per day oral dosing in advanced cancer patients. Thank you for watching to the end of this drug discovery story on IPG7236, an orally administered CCR8 antagonist. A link to the original Chem article is in the video description. Please consider subscribing to the channel, leaving a like, or making a comment. Take care.